Hi, I am Bruna. I am vet and PhD student at Federal University of Paraná in Brazil. And this research was done in partnership with Novus company. This study is about effect of dietary supplementation of essential oil and organic acid alone or combination in broilers. So one of the most effective tools to maintain the gut health in broilers is the antibiotics. So as we know, in recent years, government regulation and consumer pressure driving poultry production through AGP-free. So in absence of the AGP in feed, a several category of feeds additives include organic acids, phytogens, prebiotics and probiotics, enzymes, and immunostimulants have been evaluated for their potential to be AGP with variations of degree of success. So it's unlikely any one single feed additives will be the ultimate AGP's alternative in the multifactorial etiology of the good health challenge. The effect of the different feed additives under certain challenge will be important to help decide when and what additives to use to maintain the good health and therefore the productivity of the flock. So Novus Premium's blend technology actives ingredients protected in vegetable oils matrix and a gradual release researching the intestinal where can be effective against the pathogens. So with this, the objective of this study was to determine the effective of the protected benzoic acid, PBA, produced in feed on good histology of the broilers under commercial conditions. So the hypothesis of these studies is the PBA product in feed will improve the gut morphology, and the PBA in feed will be able to reduce the IZ score of inflammation cells in the intestinal villi. So in the materials methods, 2 trials were conducted in commercial facilities in Brazil. So these barns that were analyzed was conventional barns for almost 30,000 birds with similarly historical performance and challenge. These 2 trials were performed in different data in different years. So the first trial was conducted in 9 different barns, and was used only 27 broilers. They were divided in control group with alkanol as AGP and product with EDF on top. So the control group had 4 barns. The product group had 5 barns, and 12 broilers were used in control and 50 broilers were used in product. In the second trial in last year, 8 barns totalized 24 broilers were used for this experimental. You were divided also into groups, but in this trial, the product used the PBA was not used together with the AGP. So was used 3 barns to control and 5 barns to product, and 9 broilers were collected from control group, and 50 broilers were collected from product group. So in the both trials, 3 birds per barn, average body 2%. They were euthanasiated by cervical dislocation for ileum sampling. So for ileum sampling, we use the histology analysis, 20 villas per bird, applying the method IC inside. So the method IC site It's a method that evaluated the histological parameters in the histology. So we evaluated lamina propria thickness, epithelial thickness, proliferation of enterocytes, inflammatory cell infiltration in epithelium in lamina propria, increase of globulate cells, presence of urea, and congestion. And also, it's given for each one of these parameters a score 0 to 3, 0 no lesion and 3 the highest lesion. So also the differential of this methodology is the impact factor. So for example, the impact factor for presence of oocytes has impact factor 3 because it's known the coccidiosis cause a gut inflammation, it causes a losses of performance in the bird, and has a big impact in the bird. So on the other hand, the epithelial thickness has impact factor 1 because not necessarily these parameters are linked with inflammatory response. So for statistical analysis was used data were submitted to Shapiro-Wilk normality test and Kruskal-Wallis test. For the results, in the both trials was very interesting what happened. The both trials present the same statistical results in the same histological lesions. So lamina propria thickness, epithelial thickness, proliferation of enterocytes, inflammatory infiltration epithelium in lamina propria, when was used PBA with and without association with AGP, present a reduction of the lesions compared to control group. So this is what we observed in histology. The birds that receive the PBA in the feed was observed less lesions compared to control group. So in the sum of the all parameters that we call the easy total score, as the sum of all the parameters, histological parameters that we evaluated, we observed in both trials the same results. So there was a reduction of the all lesions evaluated statistical results between the control group and the group that received the PBA in the feed. So concluding the hypothesis, the PBA product in the feed will improve the gut morphology. Yes, we observed it. And the PBA in the feed will be able to reduce the IZ score of inflammation cells in the intestinal villi. Yes, we observed this as well, and in the both trials. So as conclusion, the broilers under commercial challenge and supplemented with PBA in diets with or without aquinolol as growth promoter improve the gut morphology by reducing the intestinal inflammatory infiltration in epithelial cells turnover, and made positive reflected in performance due to better absorption capacity and improve the gut barrier. So I would like to thank you for watching my presentation. I would like to thank Novus for the partnership and the PSA for all the support and opportunity. Thank you.